Welcome everyone to this new Gen webinar jointly sponsored by Aldevron and Synthego, entitled The Future of CRISPR Cell Therapies is GMP. I'm your host for today's presentation, Jeff Bogaliskis. I'm technical editor for Genetic Engineering and Biotechnology News, which has been covering the biotechnology and life sciences industry going on 40 years. Genome editing technologies have taken hold of the life science industry, giving rise to novel life-saving therapies not previously thought possible. The number of investigational new drug filings for cell and genome editing therapies has boomed over the last few years, facilitated in large part by the efficiency and simplicity of the CRISPR system. CRISPR's swift transition from research tool to therapeutic agent is unprecedented in translational medicine, making it one of the most important and exciting scientific breakthroughs in recent decades. In today's presentations, our panelists will discuss what it takes to make CRISPR reagents suitable for use in human therapies as these technologies move from the bench to the clinic. Now it's my pleasure to introduce you to our panel for today's webinar. Up first will be Dr. Mark DeWitt, who is a project scientist at UCLA and whose recent work has been on a gene-edited cell therapy product for the treatment of sickle cell disease, something he's going to tell us much more about in just a few moments. We'll then turn the presentation over to Dr. Robert Deans, who is the Chief Scientific Officer at Synthego. Dr. Deans has over 25 years of experience in adult cell therapeutics, including the commercialization of cell therapeutics from bone marrow. Today he's going to tell us some more about the exciting things going on at Synthego and how they could help with your research endeavors. Finally, we'll hear from Thomas Foti, who is the president of the Protein Business Unit at Aldevron. Mr. Foti is one of the original founders of Aldevron's protein service business, and over the past several years, he's been responsible for considerable growth in the business with key partnerships. Today, he's going to tell us more about Aldevron's key CRISPR services and capabilities and its collaborative efforts with Synthego. Before we begin, however, I want to remind the audience to stick around for a Q&A session that will start right after the last presentation. Don't be shy. Send in any question that you have for our panelists at any time during the presentation. It's really easy. All you need to do is click the Ask a Question tab right on the right-hand side of your screen, type in your question, and hit submit. And we're going to try to get to as many of your questions as possible. All right, with all that said, let's get on to the presentations. Mark, the Gen audience is all yours.
Hello, everybody. My name is Mark DeWitt. I'm a project scientist at UCLA and the project manager for a multi-site cell therapy project in sickle cell disease. It's funded by NIH Cure Sickle Cell Initiative and CIRM. So in this brief talk, I hope at the end of it that you'll understand how we have developed a phase 1 GMP compatible manufacturing procedure to correct the sickle cell disease, or really any other disease causing mutation, using CRISPR-Cas9 with a modular non-viral gene editing platform. To date, we have completed all 6 of our planned GMP runs, and all of them pass our prospective release criteria. We find that a plurality of the edited cells possess at least one corrected allele, which is consistent with genetic correction in sickle cell disease. We see minimal off-target editing when we use the IDT developed and now Aldevron licensed high-fidelity Cas9 variant. We see an absence of significant toxicity in vivo and robust engraftment of the edited cells in immune-compromised mice. And all of this is leading up to an IND filing very soon and a first patient infused sometime next year. So a brief background on sickle cell disease. Sickle cell disease is a devastating multi-system blood disorder that affects millions of people worldwide, including 100,000 people in the United States alone. It's a recessive genetic blood disorder that's caused by a single point mutation in a single gene, the adult beta globin gene. And that recessive nature is important to something we're going to talk about later. But sort of long story short, it's a very serious disease, but it has a very simple cause, a single mutation. Currently, the only curative therapy for sickle cell disease is an allogeneic bone marrow transplant. But access to this is limited by the limited availability of donors, particularly in African American subjects, and the toxicities and risks associated with allotransplant. And from transplant studies, we now know that only a minority of the hematopoietic stem cells need to be corrected to achieve a majority of therapeutic non-sickling red blood cells in the circulation. So this sets a relatively low bar for genetic correction in this disorder. So as I mentioned earlier, we developed a GMP compliant manufacturing protocol to correct a mutation. In principle, this protocol could be applied to any disease-causing SNP in the HSC genome. So it consists of harvest of stem cells from a patient with sickle cell disease, a brief period of ex vivo culture, delivery of the non-viral gene editing reagent cocktail, so a Cas9 protein, a guide RNA, and a single-stranded DNA donor bearing the corrected sequence, delivered to the cells by electroporation. This produces a mixture of gene-edited cells, which can then be reinfused into the patient following a conditioning regimen. This is a little more detail on the manufacturing process. So we mobilize subjects with plerixafor as a single agent because GCSF is not allowed to be used in sickle cell disease. The CD34 cells are then isolated using a miltenoclinimax instrument, cultured for up to 48 hours to stimulate the cells to divide, at which time they are electroporated using a MaxCyte GMP electroporator and the cocktail of gene editing reagents that I just described. This gives the edited cells drug substance. This drug substance is then formulated into a drug product using CryoStor 5 and frozen, and then a thawed aliquot is subject to release testing, and if the product meets all release criteria, it's released for thaw and infusion at the clinical site. So the process currently runs at between about 100 million to about 1 to 1.5 billion cells per lot. The electroporation process volume is 3.2 mil. And manufacturing outcomes are summarized here. As I mentioned, we've completed all 6 of our planned runs, 3 small scale using sickle HSPC, and 3 clinical scale using healthy donor HSPC. All runs meet all release criteria, including excellent viability, an average of 81% viability in the final products after thaw. Robust CFU formation, so the edited cells will readily form colonies on soft agar or methyl cellulose. We see correction of the sickle mutation or a surrogate site in the case of healthy donor cells of about 26% of alleles, and indels are formed about twice as frequently as the correction events. Now, of course, that is the fraction of alleles. What's happening in the cells? The cells all have 2 copies of the HBB gene after all. And so when we sequence individual cells, what we see is that a plurality of the cells have at least 1 corrected allele, which is consistent with genetic correction of a recessive blood disorder like sickle cell disease. We also see that a significant minority are uncorrected, so they would retain the sickle allele, or have 2 indel alleles. And so you might wonder, well, what are the phenotypic consequences of having 2 indel alleles? Are they beta 0, which is to say thalassemia-like phenotype, or what? And so to answer that question, we sequenced 812 individual erythroid CFU from 1 lot of edited cells using RNA-seq. And what we're looking for here is signs of beta thalassemia, specifically alterations in globin expression. So in thalassemia, you see reduced beta globin expression and a concomitant increase in gamma globin or fetal globin expression. And in the case of colonies that have 2 frameshift indels, that's exactly what we see compared to the corrected cells. So you can see here on the left that the cells that have 2 out-of-frame indels have relatively low beta globin expression and relatively high gamma globin, whereas in the cells that retain sickle or have been genetically corrected, you see increased beta globin expression and reduced gamma globin expression. And interestingly enough, we find that in-frame indel homozygotes are more similar to corrected homozygotes, and that the heterozygotes, the order of dominance is exactly what you would expect. So corrected and sickle are dominant over thalassemia. All of them have that, sickle or healthy globin expression pattern as opposed to a thalassemia one. So really, a relatively small minority of the CFU have a clear thalassemia-like pattern. And of course, we expect that the majority of those cells will not mature into RBCs, into red blood cells in vivo, because of the ineffective erythropoiesis that is a hallmark of the thalassemia. And we have some more evidence of this from mouse studies. And so the NBSGW mouse affords a limited extent of human erythropoiesis that allows you to look at whether or not you see enrichment of certain alleles in the erythroid lineage versus the entire marrow. And so what we see is that in cells that have, what we see is that the frequency of the outer frames, so beta 0 allele, declines markedly when we look at the erythroid lineage here, the CD235, and we see a corresponding increase in the sickle correction as well as unedited cells. And so what we can see here is that in the erythroid lineage, the corrected allele really comes to dominate erythropoiesis. And we believe that these results, which are consistent with clinical observations, offer a favorable profile for subjects when they're transplanted. So of course we have to talk about off-target editing. The off-target profile of this product was improved markedly when we adopted the high-fidelity Cas9 variant that was originally developed by IDT and has been licensed to Aldevron. And you can see evidence of this in GuideSeq, which is a popular technique for identifying off-targets. We see only a single off-target, which we're calling OT1. We then sequenced this and 189 additional off-targets that were predicted by bioinformatics search to confirm that really that was the only off-target, and that's exactly what we see. None of the off-targets have editing consistently over 0.1% of alleles, except for off-target OT1, which is targeted about 4% of the time. So off-target 1, it is a real off-target, of course, but it is intergenic and it's on a different chromosome. So we don't think that there is a very high likelihood of toxicity associated with editing of this site. Of course, editing is one thing, but translocations are another. And so with HBB and this off-target OT1, we wanted to understand whether or not there were translocations occurring between these two sites. And to do this, we developed a droplet digital PCR assay. It's very, very sensitive with an LOQ of about 0.01% of alleles. And we apply that to the 3 clinical scale runs. We find that we see editing over LOQ in 1 edited and 1 unedited sample from the same run. Both very low, very near the LOQ with a high error bar, suggesting that this is possibly an incidental finding. Or in any case, the translocations are extremely rare in the drug product. Another important question for the preclinical studies was the extent of Cas9 retention in the drug product. Obviously, retention could be an issue immunologically, for example, by affecting engraftment via complement-mediated lysis of Cas9-coated cells. So for this, we developed a custom ELISA assay and had to refine it extensively, but now it works very well. And when we apply that assay to the GMP runs, we see that Cas9 retention in 95% confidence interval of less than about 100 nanograms per 1 million cells, average of about 30 or 40. And the consequences of Cas9 retention will be explored in the trial subjects when we get to that point. To assess the in vivo persistence of the gene-edited cells, we injected 4 lots of cells into immune-compromised NBSGW mice. And we find that across all 4 lots, we see very robust engraftment, looking at flow for CD45, shown here in the top right. And that likewise, we see a maintenance of gene correction pretty similar to where we started, about 22.5% in the marrow, and indels maintained as well, about 58.3%. So another interesting finding from these in vivo studies was that we can use the indel alleles, which of course are highly diverse at different lengths and different locations, to understand the clonality of the xenograft. And that allows us to construct heat maps like this, where we can, rank order all of the alleles seen based on their frequency in the injected cells, and then look at their frequency in the various mice. And what you can see is that it sort of sprinkles around. Some mice are engrafted with one set of alleles, others with another, so on and so forth. And when we quantify this, we find that we can detect about 300 unique indel alleles in the injected or in vitro cells. And then once they've been through a mouse, so the in vivo engrafted cells, the average number of alleles detected per mouse is lower by a factor of about 3. So this is consistent with the concept of stochastic hematopoiesis in a small animal that can only engraft a limited number of HSC clones. But nonetheless, the graft is still polyclonal even in a mouse. If you scale this up to a human size, you get tens of thousands of individual HSC clones, suggesting that the graft should be highly diverse and polyclonal in a human subject. We recently completed our in vivo toxicology and tumor genetics. So obviously the goal of this project is to initiate a clinical trial. This will be a single dose, open label phase 1 study in patients with severe disease, starting with 6 adult subjects. If the efficacy profile is favorable, we'll consider enrolling 3 additional pediatric subjects. The first 3 subjects would be enrolled and infused next year, and the remaining 6 the year following. Just wanted to acknowledge the people that do all the work, but first the PIs. This project is led by Mark Walters, who's a physician at UCSF. Fyodor Urnov at IGI provides CRISPR expertise. Don Cohn, also a physician, has lent his fabulous GMP team and the GMP facility to this project for manufacturing. And David Martin, who is a hemoglobinopathies geneticist, has led the preclinical studies. The studies were actually conducted by Zuli Romero, the CMC lead scientist, Wendy Magis, the lead scientist for preclinical, Stacia Wyman, who's our bioinformatics scientist, Dario Buffelli, who does gene expression and bioinformatics. And originally, the Berkeley portion was led by Jacob Korn when he was at Berkeley, and now you can find him at ETH in Zurich. And so, as I mentioned, the project is funded by the NIH and CERM. We thank them very much for their support. With that, I'll take any questions.
Mark, excellent work and data, and a fabulous way to start things off. Thank you very much for that presentation. We really appreciate it. Really quickly for the audience, stick around for a Q&A session that will occur right after the last presentation. Again, don't be shy. Send in any question you want at any time. Just type in your question into the Ask a Question tab on the right-hand side of your screen and hit Submit. Okay, on to our next presentation. Bob, the floor is yours.
Hi, I'm Bob Deans. I'm CSO at Syntago, and here today wanted to give you an introduction to the company and some really exciting technology. I'm going to go ahead and shift over to a slide deck, and I'll go off video and rejoin in a moment. Thanks. Great. So let me go ahead and introduce Syntago to you. Syntago is a very interesting company. It was founded by 2 brothers, Paul and Michael Dabrowski, that were really prominent in the SpaceX organization. So they really bring an engineering approach and way of thinking to the genome engineering space. It's been something that's been very exciting for me. I've got an excellent cadre of advisors, starting in the upper left with Jennifer Dowden and moving around clockwise with Matt Porteous, Bill Scarnes, and Sir Andrew Whitty. And I really want to emphasize that the core of our organization is really around utilizing CRISPR reagents and engineered cells to build experimental workflows that can really move discovery forward and provide us with some rapid translational entry. So we're very proud of the science we do and the transparency we have. We like to educate and share our data and thoughts. And that's been something I think that's been very reinforcing across the community. One of the things I'm showing you here is the realization that we actually count our edits. And so you can see by middle of the summer last year, we actually had over 60,000 edits composed, and that was really done with about an 80% editing efficiency. And so we did that across a number of different cell types. Reason we can do that is that we use automated robotic platforms, and we can build really big statistical data sets and make some very precise and accurate measurements and determinations. So as I mentioned, we're obviously very excited about seeding the space with science and information, and we've worked in a number of different, our publications are in a number of different development sectors. So what I'd like to do here is walk you through the progression of platform build here at Synthego around the synthetic guide RNA business. And so having launched that back in 2016, we moved to be able to offer multiple guide knockout library and screening strategies. So providing guides for the researcher to use to discover and validate targets in the discovery process. So we've moved that up a notch by being able to engineer cell product and provide screen-ready cells, and particularly emphasize the iPSC as a very informative model platform, cell platform for us to utilize. And so that's really culminated with our ability to manufacture GMP guide RNAs. And I'll talk to you a little bit more about the range of offerings that we've got in that regard. And it does also open the door to point to a future that we see being heavily dominated by computational biology and machine learning tools being applied here. Really, engineering biology is the core theme of our workflow. So this is really captured more schematically on this slide. This is really about the packages we have within the discovery space around guide design, library screening, cell engineering, and the ability to utilize those to foster a targeted gene medicine, a GMP guide to be used for therapeutic use. So I'm going to show a couple more data slides to show you the type of work and areas of emphasis, and it's really done in the context of understanding what it's going to take for us to move our genome engineering tools effectively into the clinic. And so we're obviously slowed a bit by lack of precedence, by really this being a pioneering area from a regulatory perspective and opportunities just moving in. It's not unobvious that a critical component here is safety and precision of the tools we use, and being able to read and evaluate risk of cytogenetic mutation or allelic drift. And then implicit with that is the necessity of building standardized genome analytics and appropriate algorithms for analysis. And so that's really what we have embedded in the HALO platform. This is our synthetic guide RNA platform that's really visualized over on the left by the combination of biology, chemistry, and machine learning on top of these automated platforms, really giving us the ability, if you flip over to the right side, to look at very promising data outputs in arrayed screens leading to therapeutic guide RNAs. So this is really an overview of that process and tells you how broadly we initiate the process in terms of candidate guides and rapidly move through progressive criteria that include functional screening before we select our clinical knockout guide or knock-in components for therapeutic use. I want to use 3 slides and give you a specific example of how we've utilized the power of that platform. And this is relevant in the area of our COVID pandemic. And so we had the good fortune to collaborate with an investigator at UCSF, Nevin Krogan, who's shown here on the left. And Nevin had used some immunoaffinity precipitations followed by mass spectroscopy to identify the human proteins that were interaction sites with COVID molecules. So when COVID would infect a cell, structural, non-structural proteins are expressed. Nevin has identified the human proteins that interact with those in order to foster the lytic cycle of virus replication. So it was our intention to work with Nevin to identify gene knockout pools for each of these human interactome targets that could then be used to evaluate whether the virus could be impacted, slowed in its life cycle. And if so, that became a target for drug or genetic intervention. So this is a representation of the work that we've done, and Jennifer Oki here was really a star, and one person is responsible for what I'm describing top to bottom here to you. And so what we were able to do is work in a number of cell lines that were susceptible to COVID infectivity. We chose a panel of about 330 some genes, we synthesized multiple guides against each target, optimized those, and then built in multiple cell backgrounds gene knockout library pools. So this is showing you on the right our ability to recover viable knockout pools for the majority of clones that we evaluated. Obviously, some of these knockouts were in the essential genes and didn't yield functional pools for screening. The excitement here is that it really worked. And so you have to understand that we're now talking about going from day 0, 3 weeks later, and we have data back from creating knockout pools, infecting with COVID, and demonstrating log reduction in virus titer. So it's an incredibly powerful platform to do a genetic interrogation of a biology and come out with very powerful hits. And this really is what you're seeing at the bottom right of this chart, is the ability to drop virus titer by 1 to 2 logs with a knockout of this critical human protein. The other benefit here, and it's illustrated a little bit over on the left, is that we were able to distribute the same screening libraries to multiple investigators at different institutions. So our data sets came back validated by orthogonal testing and by different site use of the process. So what I'm doing here is moving to, in a couple of slides, to describe a drive from the immuno-oncology space to adopt CRISPR engineering for therapeutic T-cell products. And so on the right, I'm showing you an outdated photo of the number of companies that are active in the immuno-oncology space. And on the left, a publication from Carl June that's representing the ability to replicate the same biology by using CRISPR gene editing. And so I think we're all quite aware of the high cost of goods associated with lentiviral manufacturing and the limited contract manufacturing capability. CRISPR is a glimmer of hope here for being able to give us a cost-effective, more immediately accessible therapeutic out into the gene medicine space. So there are other drivers for CRISPR editing in immuno-oncology, and one of those is around rendering cells usable as an allogeneic cell product. The thinking here is to recover T cells from a donor, eliminate by gene editing expression of the T cell receptor locus, and then use those cells as effector cells, CAR T cells, in an immuno-oncology patient. And so this is another demonstration for the specific and exquisite use of a gene editing technology to render the therapeutic viable. And I think is really just the beginning of some exciting cell and gene therapy products that we're going to be working with. So I wanted to move towards 2 other technology components that are really effective in moving from target to trial. And so on this slide, what I'm representing is the ability, using machine learning and automated experimental approaches, to improve the editing efficiency dramatically. And that's what you're seeing over on the illustration on the upper right side. You're looking at an initial opportunity to edit using a standard guide without optimization, and seeing a standard protocol, a routine protocol, yielding about 7% editing efficiency. So very few alternate conditions tested. So what you're seeing in this figure is our ability over a 3-day period to test over 200 different editing conditions. And by iterating on those with closed loop CRISPR, our machine learning loop, we're able to bring that editing efficiency up to 82%. That's really dramatic and very powerful, I'm sure many of you realize in this therapeutic space. So underneath that is shown the consistency seen in off-target hits. And you can see across 4 different editing clones, a remarkable consistency in target and frequency of hit. So what this represents is our ability to go to the regulatory agency with a package describing our gene editing technology that's statistically robust and accurately identifies risk targets. One of the other ways we're moving to control that on-off target safety margin has been to chemically modify our guides to become light sensitive. The idea here is that we can control the duration of gene editing by shining light on a process to stop it. And so by using a very fast CRISPR editing at high efficiency, we can maximize our on-target hit. And then by shutting that off rapidly, hold that in place with very low off-target consequences. And so that's what's being illustrated in the upper left in this slide, and then also illustrated over a time course, a longer time course, as shown on the lower side of the box. So really exciting new technology that we can bring into the translational process. So I wanted to talk for a moment about our GMP guide roadmap. We produce guides in different quantities and at different states of quality to assist in the process. The research guides are done generally at low mass yield, but we do move to high yield products both in a GMP-like and in a straight cGMP format. And we can do those under varying yield conditions. So we have a number of people that are very taken with the GMP-like offering as they're moving their gene editing programs forward in development and realize the need to have a consistent reagent to meet regulatory standards and the ability to use a GMP-like material to shave a little bit of cost before moving up to a full GMP product. So that's really what's represented here in this gradient between GMP-like and GMP guide RNA. It's really driven around, primarily driven around quality control, oversight, and documentation. It's essentially the same material, the same process used to make the GMP material, but a different batch production record and quality oversight. So we're real happy to talk to people more about both the full GMP and the GMP-like products and how those can slide into the development stream. So with that, last couple of slides, I wanted to show you the overall offerings that we have in the toolbox to help you move from the beginning of guide design down through ready to use. And as well, just offer up any more time needed to contact us through our website or following the webinar for more information around the technologies. So before I turn it back over, I do want to close by talking about our enthusiasm to work effectively with Aldevron. I think they've been an incredible leader in the space of providing GMP reagents for genome engineering and other solutions. And the opportunity to work together with them to harness our GMP guide products with their ability to produce a GMP RNA is outstanding. So with that, I'm going to go ahead and stop sharing the screen, and I'll come back on and close the dialogue and be happy to answer any other questions. So really appreciate your taking the time to let me walk through the slides with you. Bye-bye.
Thanks, Bob. Really interesting stuff going on at Synthico, and we really thank you for sharing it with us. To the audience, again, one more time really quickly, stick around for a Q&A session that will happen right after the next presentation. Again, send in your question at any time. Just type it into the Ask Question tab on the right-hand side of your screen and hit Submit. Okay, Tom, I know the audience is looking forward to your talk as well, so the mic is yours.
Thank you for joining me today. And I also would like to thank my colleagues at Synthico for joint sponsoring this webinar today. I also want to thank Genetic Engineering News for hosting our program. My name is Tom Foti, and I'm the president of Aldevron's Protein Services Division. And today, I'm going to be talking to you about some CRISPR products and services and capabilities and how we plan to partner with Synthico on some of these projects. So when we think about the market, there's really a range of therapeutic application areas that we're serving. And so the clients that we are working with are doing cell therapy, gene therapy, messenger RNA therapy. They're editing cells using gene editing therapeutic approaches and also vaccines. And so when we think about the products that we need to deliver to enable our clients to have success, those are located in the second column there. So of course, we're providing plasmid DNA for cell and gene therapy, but also for vaccine work. We're making messenger RNA for therapeutic applications now, research grade to GMP, and that's a very exciting recent development. We have GMP Cas9 and SPI5 Cas9 nuclease, and also sell that at research grade. And the thing that really differentiates Aldevron is that we've really tried to focus on manufacturing basically the central dogma of biology, where DNA encodes messenger RNA and that's translated to protein. And so our capabilities from small scale to large scale and from research grade to GMP so that clients can scale with us, not just in mass, but also in quality, has allowed us to have a differentiating position in the marketplace. So imagine a world where we'd be able to produce curative cell therapies at large scale. And to get to that dream someday, we have to overcome some significant healthcare challenges. Of course, cost is something that's very important to address early on, and also quality is very important. And so the solution just might be allogenic therapeutic strategies that allow for the engineering of curative cells. And you could develop these curative cells by using gene editing techniques or an RMP, which would knock out certain genes and then knock in other genes that would be specific for the cancer that you want to address. And if you could make those edited cell populations and then be able to expand them with quality, you would be able to reduce the costs. And hopefully if you could reduce the cost, you could serve larger and larger patient populations. So in 2017, Aldevron took a bet on launching a GMP off-the-shelf solution, and we were producing the products at research grade, but the idea of producing off-the-shelf GMP was a novel thing in the industry at the time. And so since then, we have produced several batches, both at research grade and at GMP. And we currently offer the products on the left at research grade, and on the right, we have products that are offered at GMP and GMP source grades. And so we have the 1 meg and the 10 meg sizes in all 4 formats. So the whole idea here, just like I mentioned on the previous slide, is to be able to offer a research grade solution. And then when you get in maybe to phase 1, you can use GMP source. And as you move to either pivotal or commercial grade, you have a GMP solution, all with the same vendor. So I want to talk to you a little bit about RMPs, and our experience in producing the enzyme naturally led us to conversations with our clients. And those conversations often resulted in questions like, hey, could Aldevron make an RMP complex at GMP? So we would have a single reagent that would move into a gene editing event. And so here in the slide, you can see in green is depicted our Cas9 protein, and the guide RNA is complex then with the Cas9 protein to make the RMP. And so if we were going to kind of move toward a place where we could solve this problem for clients and have GMP RMPs, one of the first things we needed to do is to develop a testing panel so that we could both look at the functional performance at the date of manufacturing and do product release, but we could also, from a stability study standpoint, test products over a period of time in a way that would give us qualitative and quantitative information so that we could support our clients who were filing IND documentation. And so here we highlight the concentration assay and the residual free guide RNA and residual free Cas9 assays that were developed. So now I'm going to talk to you a little bit more about those. So one of the things we had to do was to optimize our RNP complex, and to do that, we had to develop a way to measure the functional performance. So we created a platformable assay to optimize RNP complexing, determine the linear range for cutting, and to determine the limit of detection. And I say platformable because every time we manufacture RNPs, we might be using a different enzyme complexing for sure with a new guide. And so we wanted to be able to functionally test that. And on the left, you can see our RNP complex titrated. And you can see that on the far left, it's the highest concentration where there's the most cutting. You can see that in the conversion of the linear plasmid DNA with the cut site in converting into 2 cut bands. And We also developed an in vitro activity assay that was plate-based, that's very sensitive. And again, this was helpful for us so we could determine the linear range of the assay in the EC50, which is the enzyme concentration that gives half the maximal response. The other thing that we wanted to develop was to optimize the guide RNA Cas9 ratio for the complex. And so it's important that the Cas9 was complex, but we wanted to minimize the extra free guide in solution if possible by developing an assay to do that. And so first of all, in this assay on the left, we're titrating the amount of guide RNA, and there is a correlation between the amount of guide that's added and the amount of cutting that you see. And so from left to right, you'll see that the bands on the right have a higher conversion of the linear template to the two cut bands. And so this formed the basis for us to move into more robust testing. On this slide, we're measuring free Cas9, and we developed an HPLC-based chromatography assay to be able to do this so that we could distinguish between RMP and free Cas9. So on left in figure A is Cas9 protein alone. And as you can see in the chromatogram, it elutes off at between 15 and 18 minutes. And on the center panel, we have RMP, and the RMP elutes off between 9 and 13 minutes. And so we could get that resolution between those two molecules, which was very important. And the reason it elutes off earlier is that you have the negative charge from the guide that's on the RMP complex. And so when we overlaid on the right the two chromatograms, you can see the difference in the resolution in HPLC, and you can see that separation, which is important as we started to develop the tests. One of the things we found out that's super important when you're looking at the impact of quality is it starts with high quality reagents. And it's very important that your guide RNA is purified with HPLC. And in our assays here below, it shows why. On the left, you can see a gel where lane 1 is HPLC purified, and lanes 2 through 6 are not. And you can see the smearing above and below the target. And we see the same thing when we do HPLC on the right, where the high purity guide can be resolved, but the lower purity guide has this big shoulder that creates a problem. The next thing we wanted to do is measure free guide RNA. And after evaluating several methods, we used this native page electrophoresis to find out and became basically a reliable indicator of unbound guide. And so we ran 2 identical native page gels. The left one is just a stain gel, and on the right one, it is transferred to nitrocellulose so that we could perform a Western blot. On the left, it's indicated by the arrow. You can see the lower band indicating the guide RNA, and the reason the intensity increases, the concentration increases from left to right. After transferring to nitrocellulose, we probed the Western blot with an anti-Cas9 antibody, and you can see that the antibody lights up on the Western on the top half where you have the complex molecule, but below where the free guide is in excess, it's not detecting it. So that became a helpful assay for us to determine the free guide RNA. One of the most important things that we needed very quantitative assays for that were very robust is we wanted to run a long-term stability study. And you can imagine that when you're going from research grade to tox studies to phase 1 clinical to your pivotal trial and maybe to commercial, it's a long period of time. And the client needs to have the assurance that the product is going to be stable and perform at a high level and not break down over that period of time. When we looked into the data, we found that there was little data out there to support long-term storage of RMPs. And if the goal is to qualify and bank batches of these off-the-shelf products, we needed to understand this critical parameter for the product. So what we did was we developed a stability study where we looked at RMPs stored at -24, -30, up to 6 months, and at 80 degrees, we went all the way to 36 months. So here's a little bit of the data to share. And so this is the 6-month time point, and this is our activity assay that previously I mentioned to you. And as I mentioned earlier, the RMP is titrated from left to right at each one of these different time points. And so on the far left is the activity assay from the release after the date of manufacture. And then it's -80, -24 degrees, and 30 degrees on the far right. So if you can point your attention to the downward yellow arrow, you can see that at -80 and -20, that the enzyme is cutting and converting the linearized plasmid with the target site to the 2 cut bands. It tails off maybe a little bit at 4 degrees, but we see a more significant tail off at 30 degrees, which you would expect. And so that was helpful information and kind of demonstrating to us that our assay was working. So we did the HPLC assay, and so this is data from that. And so these are results from 6 months looking at stability at the different temperatures. And so on the far left is -80, and on the bottom right is 30 degrees. So you can see what the significant difference is between the peak that you see at 80 degrees, and you see maybe a little degradation at -20 and 4, but you see complete degradation at 30 degrees. So this was good data demonstrating that the -80 stored material was stable. This is looking at free guide, and so again, we're increasing the concentration from right to left, where the highest concentrated is on the left. And then we are looking at -80, -24 degree, and 30 degrees once again. And so this is showing at the 3-month on the left and the 6-month on the right. And we were able to see again at -80, 6 months, that it was still intact. So we're happy to see that information. Moving to functional performance, this is results at 18 months. All right, and on the left is 6 months at -80, and at the right is 18 months. And so this is again showing that you're seeing the conversion from the linearized plasmid to the 2 cut bands, and so that was good to see that we could prove that both by HPLC, which you can see on the bottom, but also as functional performance. And so with that, that concludes our talk today, but I'd be remiss if I didn't acknowledge our great team at Aldebaran. And in this picture is colleagues both from Madison, but also colleagues from our Fargo site. And we had a great 10-year anniversary party, so that's what this was from. I would like to thank you for your attention today. I'd also like to thank, again, Genetic Engineering News and Synthego for their partnership. If you have any questions, we'd be happy to answer those questions. Thank you very much for your attention, and have a great day.
Thanks, Tom. Great presentation to end on, and a really great way to wrap up our webinar. For the audience, Q&A is going to begin in just a few moments, so send in any questions that you might have lingering out there for our panel. They're happy to answer any question that you might have. Just type your question into the Ask a Question tab on the right-hand side of your screen and hit submit. Okay, since we have a bunch of great questions already for our panelists, let's transition into the Q&A session. Bear with us for just a moment, and we'll start the Q&A.
All right, everyone. let's start the Q&A session. But before we do that, I just want to divert everyone's attention real quickly over to the resources tab that we have there. If you guys click on that, you can see that there's a bunch of really good data and information there from the Aldebaran and Synthico teams. But I really want to point you to the first one on the list, which is a GMP readiness assessment test that's free for everyone. If you're interested in that, feel free to go to that link that's on that page there, and you guys can have your free test, or free assessment, excuse me. Okay, so the first question is going to be for Mark. Mark, one of our audience members would like to know, for the clinical trials, do you use the same master lot for all of the reagents that will be used for the later trials once it's expanded, or do you request new reagents depending on the outcomes?
Thanks, Jeff. That's a great question. Generally for purchase of the custom, for a product like ours, where we're using a synthetic single-stranded DNA and a synthetic guide RNA, and then an off-the-shelf Cas9 protein from Aldebaran, the synthesis scale of GMP nucleotides is sufficient for all of your clinical trials, including pivotal, phase 1, expanded to phase 1, 2, and pivotal. With a protein, you might have the option of purchasing it as you go, but obviously, unless cost is a constraint, we prefer to purchase all from a single lot for the entire duration of clinical development.
All right, thank you very much, Mark. Tom, we have a question for you. One of our audience members would like to know, what are some of the technical hurdles you've had to overcome in order to launch GMP RMP as a service?
Thanks, Jeff, for that question. And so the technical hurdles were around manufacturing in QC. As most people know, producing an RMP, to go onto cells is relatively quick if you're doing it at small scale. But if you're making large scale batches, 500 milligrams, gram of RMPs, there's more technical challenges as far as managing the process control there. So that was one of them. But probably the larger technical challenge was around the QC assays for the free guide and free Cas9. So I would say those were the two main technical challenges that we had to overcome.
All right, great. Thanks, Tom. Next question, I think, is going to be for Mark, and also maybe Bob wants to chime in on this as well, too. But Mark, what kind of release testing do you recommend for the clinic? Mark, you still with us?
Yeah, or I can certainly jump in, and then Mark can add comments. This is
Sure.
Bob. Yeah. So,
Sorry, I was on.
Would you like to go ahead, Mark?
Oh, sure. Okay. So the specifications for the product were reviewed by the agency in a pre-IND meeting, and for a phase 1 product, you should have some testing for safety, purity, potency, and identity. So you can identify the product as well as measure its potency using gene editing. So identity is just the presence of the corrected allele. And then we use next generation sequencing to test for the gene editing rates in the final product. And that's pretty standard. And then in addition, because it's a HSC product, we also measure the frequency of the target CD34 cells and the formation of colonies on methylcellulose, so CFU assay. And that's those collectively, that along with the viability, kind of help define the potency of engraftment. And then gene editing, will give you some insight into the therapeutic, the potency with regards to treating sickle cell disease. It's a pretty limited release at this point. The agency did request additional assays of potency that may be required in later phase development. So if we go through phase 1, we might add additional specifications.
Yeah, it's really an exciting program, Mark. This is Bob, and I'll carry on there. I think we at Synthco really emphasize editing efficiency and the ability to hit the target very efficiently as a key parameter in safety. And from our perspective, it's really being able to apply an automated, repeated analysis of the hits and off-target hits that build the statistical case to take to the agency that you have your process under control and that your edits and your off-targets are predictable and manageable. I think over time, we'll be moving towards cytogenetic measurements against a panel of high-profile risk genes and rapid diagnostics that will start to allow us to do rapid release testing with respect to cytogenetics. We're not there yet, but that's what the quantitative and statistical database approach will lead us to. Thanks.
Yeah, and then maybe I just had a couple more comments on top of that. So we do not, the agency also requests, and we perform sequencing of the off-target site. Our guide has a single off-target, as I mentioned in the talk. And then, of course, if you have high-frequency editing of multiple sites, could be on-target, could be off-target, the agency will want to know about the frequency of translocations. In our case, we're not proposing to test every product for translocations. We have a droplet assay for that, but we certainly could if need be, and we may need to. So just to echo Bob's point about cytogenetic evaluations of the products, I think that is, emerging as an important topic in gene editing.
All right, thank you, gentlemen. Next question is for Tom. Tom, how does GMP RMP service tie into Aldevron's overall mission and strategy?
Thanks, Jeff. So Aldevron is a very mission-driven company, and we of course want to be a part of supporting curative therapies and make a big impact. So obviously that's part of our mission, and RMPs is part of that story as we talked about today. Strategically, we're a company that has been laser focused on research grade to GMP manufacturing and having options for clients as they move through that continuum of quality. And so it really fits in well with our overall plan, and so we're very excited about launching this service.
All right, thank you, Tom. Mark, another question for you. Why single-stranded DNA as a donor template? What's the length?
That's another great question. So we use, I think it's about 165 nucleotide single-stranded DNA as a donor template. And in terms of correcting pathogenic SNPs like sickle cell disease or any other SNP that one is identified, we find that single-stranded DNAs work quite well. The efficiency can be up to 50% of alleles corrected by HDR, and because you just type in the sequence to change the which SNP you're fixing, they're very modular and easy to translate. They're also very easy to manufacture. Well, they're comparatively easy to manufacture, and perhaps most importantly, because they're just a small molecule. They're a large molecule, but they're a oligonucleotide polymer. I would say that encapsulating the quality and effectiveness of the product on your C of A is much easier with a single-stranded DNA as opposed to a viral donor like an AAV6, which your C of A is not necessarily always going to accurately predict the potency of the substance. So the other alternative approach to doing this, of course, is to use AAV6 or integrase deficient lentivirus. These offer the potential of a higher efficiency, but as I mentioned, there's some issues in manufacturing and quality control that I think need to be addressed. But we're quite happy with the single-stranded DNA.
All righty, thank you. Bob, we have a question for you. What is the delay to get GMP Cas9 and gRNA?
Yeah, I'm going to answer this in 2 dimensions and then drop it over to Tom for some additional comments. I think the first thing to understand is that in producing the GMP reagents, there's a time oversight that involves quality control of raw materials coming in, your process and auditing your process and your batch production record, and then extensive analytics on the release before sending product out to customer. And so there's just an inherent delay because of quality, which is obviously critical in GMP production going forward. I think the more important point here is really to talk about factories and infrastructure. What we're seeing is a boom in the gene editing space that's moved much faster than we have infrastructure to provide the critical clinical reagents to support investigation. And so there's a limit in factory infrastructure to do this type of manufacturing. One of the points before dropping it over to Tom that I just advocate out into the audience here is to realize that very early in your thinking, if you're planning to go forward clinically, you need to line up all of your raw material providers, and you need to line up the contract manufacturing space and time in advance, well in advance of when you think you need it, in order to have a clear line of sight into the clinic. But Tom, why don't I pop this up to you vis-a-vis timing and comments of that type?
Sure. Part of the story as it relates to the time it takes from order to delivering RMPs has to do if you're going to use off-the-shelf Cas9 or SPI-5 Cas9, that's GMP, and then combine that with a custom guide, or if we're going to custom manufacture your nuclease and then combine it with a custom guide. So just as a ballpark number, if we're just going to take off the shelf, the critical path is on the guide delay, let's call it, or time that it takes to produce it as it relates to, because we can relatively quickly produce the RMP and then we're validating it for QC tests at the moment. So it could be from time to order to RMP. The simplest, let's say the quickest, maybe is 6 months, and maybe the longest would be 9 months or something like that.
All right, and Tom, stick with me. I got another question for you real quick. Does Aldebaran plan to offer sterilely filtered RMP products?
Yeah, so just to answer that slightly different, sterility will be a release spec for our GMP RMPs. So I guess the answer is yes, that the product will be released as a sterile product.
All righty, thank you, Tom. Mark, I think we have another question here for you. An audience member would like to know, what are the prospects of the dissemination of CRISPR-based allogenic therapies for sickle cell disease in developing countries where there's a high prevalence of the disease?
So that's a great question, and that's obviously front of mind for anyone who works on this, who works on translating therapies in this area. You know, the United States and Europe have maybe a little over 100,000 patients with sickle cell disease, but there are millions of individuals that have the disease in sub-Saharan Africa as well as southern India, and delivering curative therapies to these developing health systems, I think, should be a major priority for research of sickle cell disease in the next 20 to 40 years. The short answer is that it's very challenging to implement autologous transplant in these settings. And likely the future solution will be an in vivo gene therapy of some sort. However, one could imagine sort of an iterative process of getting there through first the establishment of point of care manufacturing. So this would be using end-to-end enclosed systems, perhaps even right up to the patient's bedside to harvest cells, gene edit them, and then reinfuse them. But moving on to a true in vivo gene therapy for sickle cell disease, which is kind of a different ball of wax than the one I'm working on. But I think that's probably the ultimate future. It's just there's a lot of unanswered questions as far as how one does in vivo gene editing, and you know, it will probably be some time before those technologies are ready for clinical testing.
All righty, thank you, Mark. And it looks like we have time for maybe one more question here, which I'm not sure who this is going to be directed to. You guys can tell me. The incorporation of modified nucleotides into oligotherapeutics has proven to be a critical step in vivo. Are modified nucleotides being incorporated into gRNA to enhance stability?
So this is Mark. I might start this and then kick it over to Bob. So the use of modifications improves the stability of the likely improves the stability of the guide when it's when it's inside the cells, but it's less to promote the shelf stability of the guide. And by improving the persistence of the guide inside the cells, use of the modifications generally leads to more efficacious gene editing. And so I'll just say, maybe Bob will say too, that when Synthego implemented guide modifications as a standard for all of its products, I mean, I thought that was a tremendous move, because now all of their customers, all of Synthego's customers, which is really most gene editors these days, are consistently getting the right product for the
Yeah, that's great, Mark.
Okay.
This is Bob. Yeah, and just wanted thanks to everybody. I was just going to add a little color onto Mark's comment that modifying guides for stability also includes the ability to render them with on-off properties as well, and gives you more control over the editing process. So it's really a strong point to be made. So thanks, and I'm done.
All right, thanks, guys. And with that, it looks like we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived on our website at genengnews.com for up to a year. So if you missed any parts of it, you can watch it again, or feel free to forward the link to your friends and colleagues, which we always recommend. I'd like to thank Mark, Bob, and Tom again for their informative presentations, and I'd like to thank the audience for their attention and very thoughtful questions. Don't forget to check out that resources tab with all the extra information in there. And a very special thanks to Aldevron and Synthego for sponsoring this webinar. Hopefully we'll see you again at another Gen webinar in the near future. Goodbye for now. Everyone stay safe and healthy.